A very warm welcome to the students for the part 2 of germination of seeds. So we have finished the part 1 seed germinations. So in the previous class of germinations, we have learned about definition of germinations, then we have learned the condition of germination and a few experiments to support the things which are essential for germinations. Now in the part 2, alright, that means we are dealing with the chapter seed, structure and germinations. So now we'll discuss about the 3 bean seeds experiment is the continuation of the topic germination only. Clear? Don't get confused. So now we'll discuss about the 3 bean seeds experiment. We are done with the experiment to prove the importance of air or oxygen for germination, water for germination, and suitable temperature for germination. Now we will discuss the 3 bean seeds experiment. It's in here page number 50. Clear? So let's begin. So first of all, what we require? We require 3 mature air-dried bean seeds. We are taking the air-dried bean seeds. Then we require a glass slides. Clear? So you can see over here. So you can see the 3 bean seeds. This one is the glass slides. Clear? Then you can see a beaker. So this one is the beaker containing water. So we require a beaker containing water. So requirements easy. We require air-dried bean seeds, a glass slides, a beaker containing water. So let's see now the procedures. What can be done is that. So as in the figure, mainly in the glass slides, right, we have tied the bean seeds at different positions by the help of a thread. So by the help of a thread, we are keeping the bean seeds at different positions in a glass slides, and we are putting that glass slide into a beaker containing water. Clear? Now, when we put the glass slides containing bean seeds into the water, you can see over here that the seeds at the bottom, that is this particular seeds, will be completely dissolved in water or will be completely immersed in water. So the bottom one seed is completely immersed in water. Clear? The middle one, the middle bean seeds, okay, the middle bean seed is just at the water level, you can see over here, and the top one, this one, okay, is well above the water. Okay, so what we have done? So we have taken the glass slides, and in the glass slides we have tied the gram seeds at 3 different positions. The bottom one is completely immersed in water. The middle one is at the surface of the water level, we can say, and the top one or the top seeds is completely above the water. Clear? Now we will keep this particular setup for a few days. After a few days, what will we observe? Observation. We observe that the middle seed germinates. Why? Because the middle seed gets both oxygen and water. Clear? So you can see over here that the middle seed is germinating. Why? Because the middle seed is getting both oxygen and water. The bottom seed, you can see over here, the bottom seed does not germinate or stops germinating after the emergence of small radicals. You can see over here, yeah? The small part of radical is coming over here. Okay, a small part of radical is emerging over here in case of the bottom seeds. So there will be no germination at all, but the process of germination will stop after the emergence of a small radical. Why? Because this bottom seed is not able to get sufficient amount of oxygen. It will only get little amount of oxygen which is present in the water in the form of dissolved oxygen. Because water contains oxygen in the form of dissolved oxygen. So this seed is able to get a small amount of oxygen present in the water. Clear? But the seed which is at the top, which is above the level of the water, will not germinate at all. The top seed does not germinate at all. Why? It will get the oxygen, no doubt. It is getting the oxygen, but it is not getting water. Okay, so it gets only oxygen but no water. So this experiment proves that water is essential for the process of germination, but the other things which require for the germination is not fully demonstrated by this experiment or not fully supported by this particular experiment, which is called the three bean seeds experiment. Hope it is clear for you. So what requirement? Air-dried bean seeds, a glass slides, we require beaker containing water. What is the procedure? So we'll take a glass slide, and in the glass slides we'll tie up the gram seeds or the bean seeds at three different positions. So let's say this is the glass slides. So over here we'll place the bottom seed, which will be completely immersed in water, the middle seeds which will be at the surface level of the water, and the topmost seeds over here which will be above the level of the water. And we will keep this setup in a suitable room temperature for few days. After a few days, what will we observe is that the middle seeds, which were at the level of the water, will germinate. Why? It is getting both oxygen and water. The bottom seeds it does not show any sign of germinations, or it will germinate till the radicals will emerge out. After that, there will be no germination. And the topmost seed, the top seeds will not show any sign of germination because it will only get oxygen but it doesn't get water. So this experiment, the three bean seed experiment, will actually it demonstrates the importance of water for the process of germination, but it will not demonstrate the other aspects or the other conditions which are necessary for germination. Clear? So kindly go through the page number 50 and 51. Very topic, the three bean seeds experiment. Clear? Now we'll discuss the types of germination. So we have got two types of germination: epigeal, hypogeal germination, and epigeal germination. And before discussing about the type of germination, you have to try to recall the terms epiquartile and hypoquartile, which we have done in previous classes. So kindly remember what is epiquartile and what is hypoquartile. We have discussed that thing when we were discussing about the monocot seeds and the dicot seeds. Clear? Epiquartile and the hypoquartile. So let's talk about the hypogeal germination and the epigeal germination, the two types of germination. So whenever the seeds will start germinating, the epiquartile and the hypoquartile will not elongate at the same time. At the same time, both epiquartile and hypoquartile will not germinate. Clear? Sometimes the epigeal will germinate first, followed by the growth of the hypogeal. Or sometimes hypogeal will elongate, hypoquartile will elongate, followed by the elongation of epiquartile. But both epiquartile and hypoquartile will not germinate together. Clear? So, if the epiquartile elongates, listen very carefully, if the epiquartile elongates, the cotyledons remain underground or on the ground if the seed is on the ground. Listen very carefully. If the epiquartile elongates, then the cotyledons will remain underground. Then such type of germination is called hypogeal germination formed in pea and gram seeds. So look at the figure over here. We'll try to discuss. Clear? So you can see the seeds over here. You can see the seed germinating, radical is coming out. Now over here the most important thing, you have to see the position of hypoquartile and the epiquartile. Now in hypogeal germination, the epiquartile will elongate. Alright, first the epiquartile will elongate. So when the epiquartile will elongate, then you can see over here that the cotyledons will remain underground. This is the cotyledons. This is the surface of the soil. So over here in hypogeal germination, the cotyledons will remain underground. Why? Because the epiquartile will keep on elongating. Clear? Now since the epiquartile will keep on elongating, the cotyledons will remain underground in case of hypogeal germination. But if the hypoquartile elongates, alright, if the hypoquartile elongates, then the cotyledons, look over here, then the cotyledons then the cotyledons are pushed above the surface of the ground. You can see over here. So when the hypocotyl elongate, this green portion over here, radical hypocotyl. So when the hypocotyl elongates, then the cotyledons are pushed above the ground. So the cotyledons will come above the ground, and therefore this type of germination is called epigeal germination, which can be which can be seen in the castor seed or in the bean seeds. Clear? So in hypogeal germination, the epicotyl will elongate and the cotyledon will remain underground. Whereas in case of epigeal germination, the hypocotyl will elongate and the cotyledons will come out above the ground surface. Clear? So now we will check out the difference between hypogeal germination and the epigeal germination. So in hypogeal germination, cotyledon remains underground. You can see over here, cotyledons are underground. Whereas in epigeal germination, the cotyledons are pushed above the ground. So level of the soil, you can see the cotyledon above here, over here, where it is above the ground level surface. Number 2, in hypogeal germination, epicotyl elongates faster, whereas in epigeal germination, hypocotyl elongates faster. Hypogeal germination is mainly seen in monocot seeds, whereas epigeal germination is seen in case of dicot seeds. So this is the difference between hypogeal germination and the epigeal germination. Clear? Now next one, we will see the epigeal germination in case of bean seeds, the dicot seeds. Okay, also over here, hypocotyl will elongate at much faster rate. Okay, so you can see over here the seed will absorb the water and they will swell up. So mainly from the micropyle, the seed will absorb the water, and whenever the seed will absorb the water, it will swell up. So when the seed will swell up, the radicals will come out and radical will form the root system that we know. Okay, now look over here, hypocotyl portion. The hypocotyl which grows will form a arch, ARCH. This particular structure we can say arch. Okay, ARCH, arch or loop. So whenever the hypocotyl grows, it will form an arch or a loop above the ground surface. Now slowly this hypocotyl starts or begins to straight. It becomes straight. Okay, so this hypocotyl slowly it starts becoming straight. So when the hypocotyl slowly begins to become straight, then the cotyledons will be pushed above the surface of the soil. So first it forms an arch or loop-like structure, and this loop will slowly begin to become straight. So when this arch or loop-like structure becomes straight, then cotyledons will be pushed above the surface of the soil. Clear? So therefore, this type of germination is called epigeal germination. Cotyledons become the first green leaf. We know that because cotyledons will form the green portion of the leaf. Then after that, it will soon fall off after the foliage leaf will grow. Okay, so this is called epigeal germination. Now, hypogeal germination in case of maize grain, you can see the figure over here, the maize grain. So the grain will imbibe water. Imbibe means it will absorb water. Imbibition. Okay, so the maize grain will absorb the water and it will swell up. Now when the maize grain will swell up after imbibition, after absorbing the water, then radical will come out. Radical will pass, means it will rupture. It will come out through the protective sheath called the coleorhiza. Even this we have done while doing the structure of monocot seed, that is maize grain, coleoptile, coleorhiza. So try to recall those terms back. Okay, so radical will come out. It will rupture the coleorhiza and the fruit wall, and it will form the root system. So you can see over here, radical is coming out, and radical will form the root system. So all these root system, they are formed from the radical, the future root. Now similarly, plumule will also come out. Now plumule has to pass the coleoptile, because coleoptile is the protective sheath of the plumule. So coleoptile, through the rupturing of coleoptile, the plumule will come out and it will start growing upward with the future shoot of the plant. So this future shoot of the plant is mainly formed from the plumule. Clear? Now the two produced seeds, that is coleoptile and coleorhiza, can be seen as a membranous covering on the axis of the seedling. So when we look at the seedling image, we can see the presence of coleoptile and the coleorhiza. Okay, so cotyledon in case of monocot is known as scutellum. This also we have discussed in your previous classes when we're discussing about monocot seeds. Okay, so cotyledon or scutellum in monocot seeds, it will absorb the food from the endosperm till it is exhausted. So mainly from the endosperm, this particular cotyledon scutellum or the mesh grain will start absorbing the food materials. The hypocotyle does not elongate. So only over here the epicotyle will keep on elongating. Yeah, so that's why the grain will remain underground. That's why the first type of germination is called hypoglial germination. So now we have just discussed about the three bean seed experiment, and we have discussed the type of germination, hypoglial germination and the epigial germination. And we also discussed it by discussing the bean seeds regarding the epigial germination and the mesh grain through the hypoglial germination. So over here we have finished the chapter, and in the next class we will begin with a new chapter. Thank you.